Our next speaker is joining us via the internet, via Skype or some other high-tech phenomenon. Bill Burnhans, who is a senior cancer scientist at the Roswell Park Cancer Institute in Buffalo, New York. Bill is going to tell us a very frank and personal perspective on the field of cancer diagnosis. The title of his talk, his full bio and everything and abstract is in the program, so I'm not going to spend time on it. Bill's going to speak to us about my life over and under the microscope as a cancer scientist and patient. I'm not sure if he can hear much of what we're saying, but let's give Bill a warm round of applause. Thank you.
Thank you. So, can you hear my voice? Okay. Let me start. Hold on one second. Yeah.
Yes.
I can. Okay, so I would like to start. Oh, I'm sorry. Let me know when I should start. Okay. So let me start by thanking Larry and Meredith for the invitation to speak to you today. And I also need to thank Meredith for the help in making this connection remotely. Unfortunately, I was planning on being there in person, but events developed that made that impossible. I was terribly disappointed that that was the case, but I think that the workaround that we've created is going to allow me to tell you all the same things that I would have told you if I had been there. Hopefully the weather is warmer out there in Colorado too. In Buffalo, it's pretty darn cold. I think for years around this time of the year, I would be heading off to study section meetings at NIH, and without fail, someone would always ask whether or not the snow had melted yet in Buffalo. So I can tell you that it has melted, but it is pretty darn cold. So I'm going to begin by telling you a little bit about who I am in a very general way. So I'm 67 years old. I was diagnosed with stage 3B prostate cancer in 2013. In 2014, I underwent genetic testing that revealed that my cancer was driven by a pathogenic mutation in the breast cancer gene, BRCA2. So BRCA2 and the related gene, BRCA1, are, of course, the genes that are more commonly associated with ovarian and breast cancer in women. And what's not frequently known is that they both play a role in prostate cancer as well, although the frequency with which that occurs is much lower than in breast cancer and ovarian cancer. Generally, it's about 2% of all cases. I also spend a lot of time, in addition to dealing with my prostate cancer treatment, as a patient advocate for prostate cancer patients. So I currently serve on the board of directors and board of advisors of the NCAN Foundation, which is a patient advocacy organization that helps other prostate cancer patients deal with their disease. So that's the general description. I'm going to start by telling you about my life over the microscope and give you a brief overview of my scientific training and experience. So I have a bachelor's degree in cell biology from the University of Connecticut, which I obtained in 1975. But I have to say that at that period of time in my life, I learned much more about science from my experience as a field crew member working for the U.S. Forest Service in summers of 19, whoops, hold on one sec. Okay. In the summers of 1972 and 73. So those summers I worked on a project that investigated the population dynamics of an organism called gypsy moth. And those of you who are old enough to remember might recall seeing the huge numbers of gypsy moths that infested the forests in the Northeast, especially in New England. The photo on the left is a picture of a gypsy moth caterpillar, and you can see that the leaf that it's sitting on has been chewed to a certain extent. You can see the holes below the caterpillar. And then the photo on the right is of a gypsy moth that's laying eggs or an egg mass. And in fact, there are probably between 300 and 600 eggs in that mass that it's laying. And then you can get some idea of what this was like as an infestation in this photo of egg masses that are being laid by a large number of gypsy moths. So the number of larvae that could be found back at the time of this infestation was so large that it would, I remember, for example, looking at the rear wall of our garage, which was painted white, and it looked like it was painted black, just a huge number of caterpillars. And that led to the complete deforestation or defoliation of forests in the Northeast, as you can see in this photo. The deforestation usually occurred at lower elevations where it was warmer. The tops of mountains escaped because it was colder, and the gypsy moths couldn't survive up there. So the research that we did as part of this project was eventually published, and some of the publication information is indicated in this slide. What's important about this is that this is back in, we actually did the research in the mid to late 70s. And this was at the very beginning of the time when it was possible to sequence DNA. And we were among the first laboratories to actually sequence DNA when we sequenced the genome of the H1 parvovirus. So this is a new job that I took on as a research assistant at the Institute of Medical Research of Bennington in Bennington, Vermont. I actually, before I took this job, in between my work with the Forest Service and when I returned east, I lived and worked in Colorado, down in the southeast corner of Denver, near Cherry Lake Reservoir. Back then, it was completely empty of buildings. It was mostly open prairie, and I have flown into the Denver airport many times since then and seen that it's completely developed now. So after my job at the Institute of Medical Research in Bennington, I worked for a couple of years at UC San Diego in the Department of Medicine with Mickey Goulian. Mickey did spectacularly interesting postdoc training with Arthur Kornberg before he set up shop at the Department of Medicine at UC San Diego. And I worked in his lab again on parvovirus DNA replication for a couple of years. So these are the experiences that I had that have informed my subsequent career as a scientist working over the microscope. And now I need to tell you that my family has an extensive history of cancer. And for example, my mother died of early onset breast cancer when she was 52. All 5 of her siblings died of cancer of one type or another, but it includes ovarian, breast, bladder, and lung cancer, and ovarian and breast cancer are cancers that are caused by mutations in BRCA1 and BRCA2. And so that suggested that they might have mutations in those genes, but of course, back at the time, it was impossible to determine that. My father was diagnosed with prostate cancer and died of prostate cancer treatment complications. My younger brother, who is still very much alive and actually healthy, was diagnosed about 15 years ago with prostate cancer. And a younger sister died of breast cancer at age 64. And another younger sister was diagnosed with ovarian and breast cancer and is still happily living in a town outside of Boston. So clearly, there is an extensive history of cancer in my family, and it's a history that would lead anyone who knew anything about this to run rather than walk to the nearest cancer geneticist to obtain genetic testing. And that's something that I did in addition to deciding to pursue a career as a senior cancer scientist in order to better understand the malignant forces that were killing my family and also threatened to kill me. With that history in mind, I decided that I wanted to pursue scientific training for a PhD that would allow me to work on the problem of cancer in the future. And so I went to graduate school at the University of Vermont and obtained a PhD in cell biology, working in the lab of Nick Heinz. I worked on various aspects of DNA replication, and it was a fairly fruitful graduate career in that my work was published in these two papers as well as several other papers as well. I then went on to postdoc training at the now defunct Roche Institute of Molecular Biology. I was there for 5 years. This is a photo of the institute. Anyone that lives near New York City might have seen it driving by the old Hoffman LaRoche campus in Nutley, New Jersey. It was right off of Route 3. The institute was just a really unique place. It was called Camelot in Nutley, as a title of a book that was written by Herb Weisbach, who was the director of the institute during some of the time that I was training as a postdoc there. But as he wrote in the foreword of this book, in 1967, the Roche Institute was established by Hoffman LaRoche, and that generated news coverage throughout the world. In a relatively short time, the Institute established itself as a world-class research and training center and maintained this reputation for almost 30 years until it was unfortunately closed down by Hoffman LaRoche. But as he pointed out in the book, the Science Citation Index has always rated, the Institute among the top 4 or 5 independent institutes in the United States. And I have to say that my postdoc training there was just spectacularly good. It set me up for a very productive career as an independent scientist. This is just a few of the papers that I published. Once again, most of these papers involve studies of DNA replication. So, in fact, most of my 26-year career as a senior scientist has been spent investigating different relationships between DNA replication stress and genetic instability in cancer. And so what do I mean by DNA replication stress, and how does it promote genetic instability in cancer? This schematic, I hope, reveals why DNA replication is important in the disease of cancer. Up at the top in the A portion of the diagram is a double-strand DNA molecule that has suffered a single-strand lesion. Single-strand lesions, as I think many of you know, are rapidly repaired and are not nearly as deleterious as double-strand breaks. But it's also the case that double-strand breaks can occur in chromosomes, and this can occur especially at the unwound regions of DNA at the junction of the two halves of, DNA replication forks, sorry. So when those unwound regions are exposed, then things like reactive oxygen species or radiation or topoisomerases can cause this single-strand region to, this is the only place in the chromosome where a single-strand region effectively causes a double-strand break and the collapse of stalled replication forks. So for this reason, DNA replication stress, which is involves the stalling of DNA replication forks, is really important to the problem of cancer. And why am I not able to? There we go. Okay, and that's reflected in this graph that I made a few years ago, which shows the number of citations identified by Google Scholar that link the phrase DNA replication stress and cancer over a period of several years. So back in 1999, there were very few papers, actually just a couple, that used the term DNA replication stress, but there were several hundred by 2011. And when I checked yesterday to see how many there were in 2018, it's up to more than 1,000 citations. So clearly, DNA replication stress is now recognized as an important component of the problem of cancer. So I also need to tell you a little bit about the phenomenon of synthetic lethality, or synthetic sickness. Synthetic lethality is, of course, when an organism dies. Synthetic sickness is when it is having problems remaining viable, but it is still viable. So the phenomenon was first described in Drosophila by Calvin Bridges in 1922, and then it was first referred to as synthetic lethality by Theodore Dobzhansky in 1946. This slide diagrams what synthetic lethality, how it works, in particular in the context of cancer. So you have a cell up at the top with two genes, gene A and gene B, and in the cells that are in the row just below it, there are mutations that are indicated by the stars in either gene B in the case of the first cell, or in gene A in the case of the second cell, or either of these genes is overexpressed. In all these cases, the mutations do not impact the viability of the cells. But in some cases, when mutations occur in both genes, gene A and gene B, the outcome is that is lethality for the cell. And in the leftmost cell, this involves mutational changes to the structure of both gene A and gene B. And then in the middle cell, with the designation C, one of the genes, gene A, is mutated, but then the second gene, gene B, the function of it is blocked by the use of an inhibitor. And so this basically involves pharmacological inhibition of the function of gene B that introduces synthetic lethality when there is a second gene that is mutated. And so that makes it possible to use pharmacological inhibitors that can be used to treat cancer, for example, in patients that have mutations that are that can be targeted by specific drugs, and that can result in the specific death of tumor cells. And then in the panel labeled D, something similar can occur when one of the genes is overexpressed. So this is this type of interaction between pharmacological agents and drugs, mutations is I'm sorry, hold on one sec, is the basis for much of what is referred to as personalized medicine, where information about the about the mutational status of a patient or specifically of their tumor cells can be used to direct the treatment with specific compounds. So this is something that we actually, my laboratory that is, employed, we described actually first in a book chapter that we wrote for a book titled DNA Replication in Human Disease that was published by Cold Spring Harbor Laboratory Press in 2016. This chapter was written by myself and Tony Carr and Jeff Wall. And in this chapter, we described basically how this approach could be used to treat cancer patients. And towards the end of that chapter, we described the synthetic lethality therapeutic approach, employing drugs that act synergistically with replication-related and other mutational defects in cancer cells to specifically target these cells for elimination. And at the time, two examples of this approach that were commonly cited were the use of G2 checkpoint abrogating drugs like 7-hydroxysterosporin, which is also known as UCN01, which inhibits the checkpoint protein CHEK1, and that can be used to selectively eliminate cancer cells that have accumulated DNA damage during S phase due to G1 checkpoint defects. It's also the basis for the increased sensitivity of BRCA1 and BRCA2 defective cancer cells, as I mentioned before, specifically to inhibitors of the protein poly-ADP ribose polymerase, or it's also known as PARP. This was the basis for experiments that we performed back in the mid-1990s that involved screening panels of yeast mutants. So specifically S. cerevisiae, which is budding yeast. These are the yeasts that are used to make beer and bread. And at some point in the 1990s, NIH was encouraging cell biologists to use S. cerevisiae and another yeast called Schizosaccharomyces pombe, or S. pombe, as model organisms in their research, and they provided funding for that. And so my laboratory switched from doing experiments exclusively in mammalian cells to also doing experiments in budding yeast. And the experiments that employed this approach were published in another book chapter that we published with Joel Huberman, who was one of my colleagues at Roswell Park for years, And when we screened this panel of about 100 hypomorphic mutations or mutational alleles in S. cerevisiae, what we found was that those that harbored mutations specifically in genes that encoded proteins required for initiation of DNA replication were super sensitive to the drug. And eventually we determined that the drug that we were studying, which was a drug called adazelicin, had specific effects that involved inhibiting initiation of DNA replication. Lee Hartwell and Jeff Wall, and as well as a few others, were at a symposium, a Pescolar symposium in Italy that I attended and gave a talk about this, and we had a very vigorous discussion of how this might be a useful approach to drug discovery. In fact, this was about the time that Lee Hartwell and Stephen Friend were launching the Seattle project out in Seattle, which used a similar approach, except that they were screening a much larger panel of mutants than the 100 or so that we were able to obtain for our studies. So, so that pretty much summarizes my life over the microscope. And my life under the microscope begins with my diagnosis, as I told you earlier, of stage 3B prostate cancer in 2013. And just a brief overview of the treatments that I underwent after I was diagnosed. They included androgen deprivation therapy, which is a standard treatment for prostate cancer. And that is ongoing 6 years after I was diagnosed. I went through 45 fractions, meaning 45 treatments of radiation therapy in 2013. I also underwent immunotherapy with a drug called Provenge in 2015. And then finally, I was also treated with a drug called olaparib, which is an inhibitor of the PARP protein. I also underwent a different type of immunotherapy called Keytruda in 2016. And in 2018, I was treated with a drug that inhibits a protein called ATR, or ataxia-telangiectasia related. This was a compound that actually targeted a genetic pathway that my laboratory discovered back in 2003. It's a constitutively active checkpoint pathway. And my treatment with this inhibitor occurred as part of a clinical trial at Dana-Farber. And it unfortunately, it was working pretty well, but it also did a number on my blood cells, both my white and red blood cells. So we had to stop that treatment. And then now I'm being treated with conventional chemotherapy, including at first docetaxel, and now cabazitaxel and carboplatin. And the good news is that they are actually working well to knock back my disease. So as I said, in 2015 or thereabouts, I think I may have used a different date in an earlier slide, genetic testing revealed that my germline harbored a pathogenic mutation in the BRCA2 gene. And as I said earlier, at the time, there were very few urological oncologists who were aware of the fact that BRCA2 and BRCA1 were known to play roles, at least in a small proportion of cases, of prostate cancer. And So as some of you might know, BRCA2 plays a role specifically in homologous recombination mediated repair of double-strand breaks in DNA. And that means that it operates in a genetic pathway that my lab, like the ATR protein, has investigated for dozens of years. And so defects in BRCA2 and BRCA1 make cells susceptible to the class of drugs that inhibits PARP, as I said earlier, via a mechanism that actually involves synthetic lethal interactions. And that's depicted in this slide here, where normal cells with a double-strand break can be repaired with a functional, if they have a functional BRCA protein, and if they have functional PARP1. And then when cells have a mutation in BRCA1, but PARP1 is functional, DNA repair can still occur. And then in panel C, cells that have functional BRCA proteins, but do not have a functional PARP1 because PARP1 has been inhibited by pharmacologically, are still able to repair double-strand breaks. But then finally in panel D, cells with BRCA mutations and that are inhibited with drugs, in which PARP1 is inhibited with drugs, no longer can undergo DNA repair, and that results in cell death. So that approach was used to great advantage in my case. In fact, as a scientist with expertise in DNA repair pathways, I've played an unusually active role in planning my treatment. And that is actually described in this article that was published in the New York Times Magazine back on May 15th in 2016. So basically, what it describes is how when most patients are sitting in exam rooms getting advice or information from their oncologist, they're busy scribbling notes as he or she talks. And in my case, it was the other way around. My oncologist was scribbling notes as I described to him what BRCA proteins were all about and what the drug olaparib could do for me. And I asked him to put me on olaparib as treatment. And it worked for almost 3 years. So it bought me that much time. It knocked back my disease to almost nothing. But then eventually it stopped working, unfortunately. And this is something that frequently happens in cancer treatment. And we now know, based on sequencing of tumor cell DNA from a biopsy that was performed at Dana-Farber last year, that the BRCA2 mutation in my tumor cells anyway had reverted, meaning that the BRCA2 protein that it encodes has resumed wild-type function. So my tumor cells are no longer sensitive to olaparib or other PARP inhibitors, which means that I needed to find other types of treatment. And as I indicated earlier, that included treatment with the inhibitor of the ataxia-telangiectasia related or ATR protein, which functions in the pathway that my laboratory discovered. And now I'm being treated with conventional chemotherapies, as I said, that are doing a good job of knocking back disease. So I guess at this point, I'd like to thank the clinicians that have been carrying me along to this point. That includes a terrific PA at Roswell named Marcus Sikorsky. Kamal Chada is my oncologist at Roswell, and I am just so fortunate to have him as a oncologist. Jim Moeller was my oncologist at the beginning of my treatment. Jim is was anyway, the chair, he may still be, I'm not sure, of the committee run by the National Comprehensive Cancer Network that establishes the standard of care for prostate cancer treatment throughout the country. And he's been terrific to have on my team as well. Roberto Pele was another clinician on my team early on until he left Roswell. And then finally, Atish Chowdhury at Dana-Farber has been just terrific. And he's going to be putting me on another clinical trial, hopefully within a couple of months, that will involve treatment with a drug that is able to home in on tumor cells based on its conjugation with a protein that has an affinity specifically for prostate cancer cells. His assistant, Monica, he has been a huge help. And then something that I didn't tell you about was that about 2 weeks after I was diagnosed with prostate cancer, I discovered a lump in my quadriceps that turned out to be another tumor, unrelated to prostate cancer. Fortunately, it was a benign tumor, and Dr. Singer at Sloan Kettering excised that one. And then finally, Johan de Bono is a very well-known expert in prostate cancer and treatment with drugs that cause synthetic lethality, including drugs that inhibit the PARP protein. And he's been really wonderful in providing advice to me by email and phone calls over the last 6 years. Okay, so with that, I'll entertain any questions that anyone might have. I'd also like to point out that I hope that my talk, that at least some of the members of the audience who are postdoctoral fellows or graduate students will understand or take the success that I've had in various treatments as evidence that the work that they do on the bench in drug discovery, such as the work that the graduate students in my lab have performed over the years, really can provide, can end up providing drugs that make a big impact on the lives of cancer patients. So I hope that those of you who are students or postdocs will keep that in mind and not lose time as when your experiments don't work, for example. So thank you very much for your attention, and I'll answer any questions that you have.
Thank you. Thank you, Bill, for an extraordinary and inspirational story. I'm sure we have some comments or questions from the audience, and if you don't hear them, I will relay them to you.
Okay.
Hi, great to hear your voice and wishing you well. My question is, you said that you had a reversion to wild type, if you will, shorthand, for the BRCA2 gene, and I wonder if you have any thoughts about the genetic mechanism by which that might have occurred.
Sure. So can you hear me?
Yes.
And you can hear my voice? Yes. Okay. So the mutation that I originally had in the BRCA2 gene was a mutation called a frameshift mutation. And the reversion mutation involved the addition of a nucleotide to the sequence of the BRCA2 gene that restored the in-frame reading of that gene by the cellular machinery. And so it became, it basically turned it into wild type again. So that's basically the mechanism. It was a frame shift mutation that destroyed the frame shift, the wild type frame that the gene was read in, but then eventually another mutation restored that frame. Does that make sense? Oh, and why did it happen? I have to tell you that I also need to thank my wonderful wife, Deb, who is a computer scientist and has been helping me with my talk over the last few days. So your question was?
Is it from PART? Aren't they responding to PART?
Oh, yeah. Okay. So Deb is reminding me to say that it probably reverted because of selective pressure from my treatment with a PARP inhibitor, which just, made it more likely that this mutation would occur.
Thanks, Bill. This is Chris. This is more of a layman's question. Although I did have quite a few family members that have died of cervical, prostate, and those types of cancers. I had a question regarding oxygen therapies or naturopathic therapies. I know it's maybe not as popular as where you may live, but in Boulder, naturopathy is more accepted. I guess it's more of a calling than maybe a specialty as molecular genetics or the art, as maybe it's called. But anyways, my question is going to with the, and maybe that might be brought up later today, another discussion, but as far as cancer being, I guess, anaerobic, in other words, it doesn't like a high oxygen environment. And I was just wondering if you had experimented with that at all. With myself personally, I've been using oxidative therapies to help break down fibroblast or scar tissue in my liver from having a Hep C infection. But it's not really breaking down the collagen, so that's another modality. But I was just wondering, I'd heard a lot of great things about that, and the side effects are pretty negligible. So anything you might want to share. So thank you kindly for your time, sir, and good luck with your treatment.
Okay, so I had a great deal of difficulty understanding.
He wants to know about
But-
oxygen therapy.
Really,
well, hold on. My wife might be able to help me with it. Hold on one second. Then, yeah.
No, I just want to say that actually considered oxygen. Hear us?
Yeah.
That was offered to him, and we spent some time in school. Remember that?
Yeah.
And there's reasons why he decided to do this.
Yeah, okay. So we did explore it as a potential therapy, and we decided not to pursue it. And I'm blanking on what the reason why was.
Well, I think there are a few things. I think you had other more promising things like the PARP inhibitor at that point.
Yeah, that's actually very true. Yeah.
You
And-
just tell them that.
Yeah, so we decided to, the PARP therapy was clearly very promising, and we decided to go in that direction at the time. And since then, to tell you the truth, I haven't gone back to think about how that might also be a useful approach. But now that you're reminding me of it, I think it's something that I will pursue in the near future. I mean, clearly that is also a very promising approach. Does that answer your question? I hope.
I want to thank you for your detailed description of quite a journey, and wonder if you could comment on the effect of your deep scientific experience and intellectual rigor over decades that led you to become this consultant in your own treatment room. The effect that had on the emotional processing of your diagnosis and prognosis, having witnessed your family and so many other people go through the ravages of cancer,
Okay, so what I can tell you is that my background as a scientist has made it possible and actually easy to look on everything that is happening to me with a much more objective eye. It's much more, there is a certain amount of emotional distance between myself and my treatment that I don't think that other patients probably would be able to have without the background that I do. And so that is, actually very valuable. I'm always, focused on the intellectual aspects of my treatment and not the impact that it has on my, emotional state. Oh, my wife is, hold on one second.
That's an issue.
Oh, okay. Yeah, my wife is reminding me that doesn't mean that the emotions don't become involved at different points in my treatment, and it can be quite difficult. It's it's not a good state to be in. But on the other hand, it often works out to my advantage in ways that I never would have expected. I can tell you, I'm a little bit reluctant to, to tell you all the details, personal details of my case, but I think this is probably important. I was diagnosed about 2 months ago with a thrombus in my vena cava, and so this is, it's a blood clot, and it could have broken loose at any point, and it would have killed me very quickly. And there was nothing that I could do about it other than undergo treatment with anticoagulants, which was the case over the last 6 weeks to 2 months. And 2 days ago, I learned from a CT scan that thrombus is completely gone. So the anticoagulant therapy worked, and I can tell you that was a cause for great joy in our household when I learned that was the case.
So your reaction to that was different from cancer because you're not an expert.
Oh yeah, my wife is reminding me that I should say that my reaction to that was quite different than it was, if it had been something more directly related to cancer, because it's something I'm not an expert in.
So, Bill, thank you very much for that inspirational talk. Your patient journey has taken place over a time where we've had the appearance of new generations of drugs, and obviously you've tried new therapies as they've come available. If you step back, can you make any generalizations about the right sequencing of these different types, these different generations of drugs, if they were all there, so to speak, at the beginning?
That's a very good question, and I don't think that there are any good answers to it. The sequencing that I underwent is actually quite different than most patients who are undergoing therapy with PARP inhibitors, for example. I think that most patients are being treated with these drugs after they've gone through conventional treatment with conventional chemotherapies and other drugs. And in my case, it was relatively early. I'm not sure, from my own experience, I'm not sure that how much of a difference it makes, at least some of the time. I mean, I can imagine that there are cases where the sequencing really does make a big difference. But I don't think that it is making much of a difference in my case, and that might be the case for other patients as well.
Bill, this is Larry. Can I want to ask a follow-on, I think.
Sure.
Does the fact that your BRCA2 is now pseudo wild type again mean that you may end up becoming eligible and helped by Keytruda, the thing that didn't do so well for you before, now that you have a different kind of tumor? Is that possible?
Yes, it is. And that is also something that I will be pursuing. The caveat here is that I've been treated with Keytruda 2 times. One for a brief period of time back when you and I were discussing this. And I was treated for about 5 weeks, I think. And it really didn't have much of an effect until the very end of that time period. And then it appeared to be having an effect. But then I went back on Keytruda a little less than a year ago, or just about a year ago. And this time it did not have, the response that I had was, if anything, was more related to something called pseudo-progression. It appeared that my tumor was actually expanding in size very quickly. And that also is something that happens occasionally with patients that are treated with Keytruda. So we didn't give it enough of a college try to know for sure that, that's the answer. And when I'm finished with my current rounds of treatment with carboplatin and cabazitaxel, I want to try Keytruda again and see if I can get some more mileage out of it. I really hope that that's the case. As you know, Keytruda is a miracle drug for many patients. And so I hope it will turn out to be the case for me.
Well, Bill, thank you very much. I think we should leave it there, but the whole audience here has been moved and will remember this talk for long after they leave this auditorium. So thank you so much and best wishes in your continued treatment.